Yep.
Good. 3, 2.
Hello and welcome everybody. Today is January 11th, 2023, and this is the Soil Food Web Movement, Webinar 1, Understanding Soil Health. I'm your host, Brian Vag, and before we get started and let folks join, in the chat window, there's a button at the bottom of your Zoom screen, if you can go ahead and just put in where you're coming from. We like to see where all of our attendees are around the world. And I can see some of you are already experienced because before I asked, you already put in where you're coming from, so that's great. And oh boy, it is just streaming in. We've got all over the place, man. It's happening so fast, I can't even read these anymore. We've got, I mean, really, I think we just pretty much hit every continent within the first 10 seconds. This is crazy. So it's great to see that we've got quite an international audience, and thank you for all you folks that are joining in some kind of weird time zone. I suspect some of you, it's probably 2 in the morning or 3 in the morning for you're out, and you're out here taking part of our webinar series. Okay, Adam, if you can go ahead and move to the next slide. So we want to tee up what the Soil Food Web Movement webinar series is going to be for January, and I think we've got a great lineup for our January promotions. Today is webinar 1, which is Understanding Soil Health, and we're really going to take a look at how the soil microorganisms form the symbiotic relationships. And we're also going to hear from one of our consultants and some of the work that he's doing out there in the field. And then webinar 2, which is going to be on Wednesday, January 18th, we're going to be talking about the Soil Food Web community and really talk to some of the consultants that are doing the good work out there and helping make those transformations using the Soil Food Web practices. And then in webinar 3, which is going to be on Thursday, January 26th, we're going to get into some success stories. In fact, it's going to be one of my clients, Jen Serp, from Sonotera Farms. And so she's going to be talking about the work that she's been doing on her farm and some of the successes and also some of the challenges too that we've had with her farm. And then in webinar 4, we're going to get to more success stories. This is going to be Grass Valley Farms in Montana. And Casey Ernst, one of the consultants, is going to be working with or speaking with his client, Corey Miller from Grass Valley Farms in Montana, and looking at the good work they're doing out there as well. So this should be a fantastic January series. And want to cover some rules of engagement before we actually get into the meat and potatoes of this webinar. First of all, everybody will be muted. There are so many people that are attending. We want to make sure that we cut down on background noise, so everybody will be in a muted mode besides the panelists themselves. And then we also have, we'd like to hear questions from you. And so you can see at the bottom of your Zoom screen, there's a Q&A button, and you want to click on the Q&A and post your questions. And again, these are going to be questions that are going to go to the panelists. But there's also the chat window, which a lot of you are already filling in where you're coming from. Go ahead and use that to chat amongst yourselves. We always have some good dialogue happening in the chat window as the webinar is going along. And just remember, if you have a question, put it in the Q&A section, not in the chat window, because more than likely, if you put it in the chat window, it won't be ready for the panelists to be able to talk about. And then really enjoy. We hope that you have a really good time going through our webinar today. So let's just talk about a little bit the timeline and our topics. So we're going to go through introductions, which we're doing now, and then we're going to get into Meet the Microbes with Dr. Adam Cobb. And then we're going to talk about ecosystem functions with Dr. Elaine Ingham. And we're going to hear from Ronald Flores going through soil biology in action, and he's going to go through a tobacco case study. And then we really have a good amount of time for Q&A. So we have about 45 minutes scheduled for Q&A. So this webinar should roughly be around 2 hours today. All right, next slide. And I want to introduce the panelists. And so I'm going to have each panelist introduce themselves. And we're going to start off with Dr. Elaine Ingham. Elaine, I hand it over to you.
Okay, I just had to figure out where the unmute button was actually. So I'm Dr. Elaine Ingham. I'm the crazy person who started all of this because I'm working with people in a number of different institutions of higher learning, put together this whole food web story and how the different components interact with each other and do some really amazing things. And most people had no idea that these organisms in the soil actually did anything useful for your plant. The attitude generally was that all these organisms in the soil were diseases and pests and problem organisms, and the only good bacterium or fungus or protozoan is a dead bacteria or fungus or protozoa, when in fact it's the exact opposite. So getting people to understand the importance of all of this, and the presentations today will really help you understand why it is that these organisms in the soil are important, as well as ways of getting those organisms back into your soil once those organisms are destroyed by too much tillage, or by the use of inorganic fertilizers, or by the use of any toxic pesticide or herbicide or fungicide, etc. So I hope you'll enjoy the whole lineup we've got for you today.
Thank you, Elaine. All right, now we'll hear from Ronald Flores. Ronald?
Yeah. So good morning, good evening, good night. I don't really know. I'm really happy to be here sharing that time with all of you. Back in 2015, when I first heard about the soil food web, it blew my mind. And somehow the challenger part of myself said, is that really true? So I'm going to check it out. And now it's more than 5 years that I'm doing it. And I got hooked. And on the road, I just took the whole classes, and then the compost and the microbiology, the microscope, and and it's very naturally that I envisioned how to actually make those transition happen, which I did. So now I have two companies, one in Europe and one in Peru, doing the same things, and we are really at the forefront, really making the job on the film, working with farmers, teaching them, challenging them. They challenge us, and this is very fun. A lot of human drama and microbiology. This is what I do. I'm really glad to be here.
Thank you, Ronald. All right, Dr. Carla Portugal.
Hi, everyone. It's always a pleasure to be here interacting with every webinar. We have more and more people, so it's super energizing. I'm an environmental scientist, and work with Dr. Elaine just helps me in the whole school. It helps me to fulfill a lot of boxes and really have translating all the theory into actions and see the amazing results that all our students and alumni and consultants like Reynolds are bringing to us on a daily basis routine, just keep us inspired and energized to continue the work. So thank you everyone for joining us today.
Thank you, Carla. All right, Dr. Adam Cobb.
Yeah, thanks for the introduction, Brian. So I'm a content creator and science communicator here at the school, which really means I get to have the fun job of sort of jumping around to a lot of different projects. And many people are aware of the foundation courses, and that's been a big focus of mine in the last several months. But I get to come on webinars and get to have one-on-one meetings with Elaine where we talk about the future of the science of the school and where it's going. And so it's just it's kind of like, I think my unofficial title is just the soil nerd, right? I'm just a soil nerd, and so I'm dancing around to all these different things, but so happy to be here with y'all today.
I think Grandmaster Soil Nerd. You're up there in the soil nerd hierarchy. I am Brian Vag. I am a soil food web consultant. I think my story kind of mirrors very similar to Ronald. Once I found out about the work that Elaine was doing, I had to know more. And as I started to know more, I was like, this is something that I really, really want to do long term and help people make that transition from growing with agrochemicals or kind of traditional organic and moving into growing with biology. And it's been quite a ride and super fulfilling in doing the work that we do. So I'm happy to be here to be able to be a host, and also happy to be part of the community doing this kind of work. So without further ado, let's go ahead and start off on our content. So Adam, I'm handing this over to you.
All right, thank you so much. So I get the probably the funnest job today here in the webinar of just talking about these microbes. these microorganisms in the soil and what they do, and putting together, kind of translating, or just putting my own little flair to the really amazing thing that Elaine and other researchers around the world have woken the world up to in the last several decades. But I'm a big picture guy. And so I like to get into some position where I can look down and say, what is the trend of the whole world? So when we're talking about soil health, unfortunately, I think we need to grapple with the reality that globally, our soils are in trouble. And it would be ridiculous for us to ignore this and pretend that this isn't the case, because this is a threat to our way of life, right? So this map, produced by some researchers and the Food and Agriculture Organization of the UN, has been part of this. This is something I go to almost all the time to start this conversation about soil health, and say we have to recognize that the areas in red and pink are areas where there's been massive degradation of soil health in the world. Loss of soil carbon, loss of productivity. By some estimates, based like on a historic analysis, our soils are 30% less productive today, right? And this is driven primarily by the way that we have used and abused them, especially in, so-called modern agriculture, kind of post-World War II agriculture. But even historically, some of these areas that are in white here, where we either don't have data or we say that it's completely bare, these are just like the cradle of human civilization. What happened? How did these areas that helped, I should say Western civilization, right? Mesopotamia. We know that there were these robust ancient groups like the Babylonians here. And then there was the loss of their productive soils. And this is a thing that's repeated through European history, through Roman antiquity, that when the soils are degraded, the civilizations fall. And, when there's a lot of different ways to sort of explain soil health, I think some things are very intuitive. Living things have health. So I like the term soil health. Some people fight about it, but I like it because it reminds me that soil is alive. But if we want to get technical, there are things that we can measure. And again, the global trend here is that in terms of the services, the things like cleaning water, or cycling nutrients, or pulling carbon from the atmosphere, or providing food, fiber, and fuel to humanity, those functions, those key services that the soil and really the soil life, the microorganisms, help us to appreciate or enjoy every day, we are at a tipping point where if we look ahead 30, 40, 50 years and extrapolate the damage of the soil forward, it's going to look really rough for humanity. And we have to do something different. That's why I'm so passionate to be a part of the school and this movement to regenerate soils, because I don't want to live in a world where there's 2 billion human refugees that are fleeing the loss of their way of life, right? Because their soils are worn out and unproductive. So that's the big picture, and it sounds scary. I'll get to the hope later. Hang on with me here.
Adam, I'm sorry to interrupt you. Is there a way you could move your microphone a bit closer or something, because the volume's a little bit low?
Okay, I'll lean in this direction here. So I don't want anybody to miss it as we make this transition from the big picture to the more concrete. So at a farm level, at the level of a garden, what are the things that we could measure, that we could examine, that we could assess, that would help us understand that our soil's check engine light is coming on, right? What are the key indicators? And likewise, what are the things we can examine that can really tell us there's health here, we're going in the right direction? Sort of the way that a doctor might look at your blood pressure, or do a blood test and see where your various hormones are at in your body, as a way to say, are you inside of the range of things that we consider healthy.
Can I butt in just a little bit?
Please do, Elaine. Always.
And one of the things that I want to emphasize is that we have methods of assessing what the problem is, and also how do you fix that problem. We want those assays to be something that growers can get their answers in a 40-second period of time instead of having to wait 40 days. So we want all of the things that we're doing to be highly practicable, highly useful for the average person growing plants and trying to make a living that way.
Oh, I'm so glad that you said that, because, when I was in grad school, I was using some laboratory procedures that I'm like, who's going to pay $200 per sample to get this? And what does this information even show us? And so one of the things when I came here a little over a year ago now to the school is I said, Dr. Elaine has done it. She's found something practical that can give you a look at the holistic ecosystem of the soil. And you can start to say, what's missing? Why is that missing? What practice have we used on the management of this land that has caused this problem? And it really comes back to, these are not the only microorganisms or important organisms in soil, but these are 4 very good indicators for us. The bacteria, the fungi, the protozoa, and the nematodes, when we take a sample of soil and dilute it and put it on a microscope slide and look at it with good methodology, right? The kinds of things that we're training people to do with microscopy here at the school, we can actually see that hidden jungle that's going on in the soil, right? Or that hidden forest, where I could walk around in a forest, like a real forest, a large forest in the Pacific Northwest or something, and I could say, why are there no birds? Something's wrong here, right? It's hard to do that with soil because it's microscopic, because everything is tiny. So Elaine has figured out.
And you have this big problem called soil. It's, it hides everything. You really have to know how to dilute that sand, silt, clay, rocks, and pebbles into something where you can see the organisms. And that's a really important understanding when you're trying to assess your biology in the soil.
Absolutely. Because if you put that soil sample diluted properly, done with the right methodology, on a microscope, and you say, I don't see even one filamentous fungus, I should say the singular, in here, or I don't see any protozoa, or the only nematodes I see are the kind that I know that hurt plants, right? Because there are nematodes that eat plant roots, but there's a lot of nematodes that are very beneficial, right? It's another example of something that's been kind of demonized in the mainstream, like, oh no, nematodes. Whereas actually, if you were to go to a jungle and say, there's no tigers in here, you would say something's wrong. Well, nematodes and protozoa are like those top predators. We need them there. And I'll get into a little bit more detail as to why. Let's start with the big picture again with the soil food web. The life that we're all accustomed to is based on a bit of magic. That when sunlight energy hits plants, the plants have the capacity to turn that sunlight energy into sugars. And they need some of those sugars to build their own tissue and to make other things like starch, the things that we harvest from systems like grain. But they also share a lot of those sugars through their root systems with the microorganisms in soil. And that's because the microorganisms actually end up driving the health of the plant. It's willing to give up some of its precious resources because in exchange, through its relationship with fungi, bacteria, nematodes, protozoa, arthropods. I mean, we could go on and on, right? Through these beautiful relationships, this symphony that's going on in the soil. Even if we said like the plant is kind of a conductor, everything is important. Everything is working together. These systems have developed over millions of years to optimize what's happening with the photosynthesis of the plant, with its nutrition. And so we can also gain the benefit of eating plants that have such a intact, healthy relationship with their microorganisms that the things we're eating have more nutrition. They have more, just in general health, so they're not prone to being attacked by pests and diseases the same way that a sick plant would be. And so, I'll go into these relationships a little bit more, but it's visualizing the way that this is a community. This is a beautiful, a cooperative and collaborative situation. Yeah, there's some things in there that maybe are a little antagonistic too. We can't forget about those, but we can keep them in check. If the whole system is working the way that nature has developed it, then your root feeders, your pathogenic organisms are going to be managed by all of these beautiful relationships that are going on. And what we're finding and what you'll learn about in this series is that this is not just some nerd stuff. Farmers who are utilizing this are increasing their profitability. In fact, on Monday, I met a farmer from South Dakota that talked about, having soil compaction at this depth in his soil 5 years ago. And then when he met Elaine and started using some soil food web methods, the compaction cleared up, and now his plant roots could grow that deep. And now they could grow this deep. And now he can walk around his field and push a penetrometer all the way in and not hit a compacted layer. Because he replaced the wrong soil management, the soil management that was degrading the health of the system, with better soil management. And he got his microorganisms back in his system. So it's not just save the planet, it's also save the farm, save the business, right?
Okay, let's keep having food that's got good nutrition in it. That's the best thing.
I tend to want to eat at least a couple times a day. Mm-hmm. So I'm a fan. So this is the metaphor that I've been reaching for to explain, out there in the world of regenerative agriculture, in what the university systems are producing for research, there's a lot of important ideas. How we breed plants is important. The way that we, the timing of our cropping system, other ways of managing the whole farm, even mechanization, which is something I don't know that much about, but on a large scale, how are you using machines to help? But the reason I care about what we're doing at the school is that there's this term we call the keystone, the top piece of an arch that we're building. And if that keystone is missing, the arch falls down. So the soil food web is that keystone. Anybody who's doing any agricultural practice, who's reaching for any regenerative tools, if they're ignoring, and that includes not assessing, not actually looking, observing what their soil food web is like, they're missing the key piece of the recipe, the key component, the keystone of the arch. So we're not the only ones who are recognizing the connection between soil and food. Many people know Paul Stamets. But I have to say that when Elaine got started in her career in academia, people were like, microorganisms, right? Or like she said earlier, like, oh, those are the nasty things that we're trying to sterilize out of our system. And the beautiful thing is that she realized, wait, if we got enough of these beneficial fungi, bacteria, protozoa, and nematodes together, they would do the essential work of keeping our system healthy, balanced. And as a result, we get healthier plants, and it all connects, right? It's holistic. So who are these key players? Well, the bacteria and fungi, naturally, they're trying to find their own, they're trying to make their own living in soil. They're trying to produce these compounds to break down organic matter. They're trying to pull nutrients out of the clay, sand, silt, pebbles in the soil, and put those nutrients in their own bodies, and also build the structure of the soil. So using glues and the physical entanglement, the bacteria and fungi are altering their habitat. They're looking for food, and they're making a house for themselves, right? And then along come protozoa and nematodes that are more like the lions and tigers and bears of soil.
Oh my.
And they start chowing down on these bacteria and fungi. Thousands of bacteria a day can be consumed by a single amoeba. So as they eat them, they are releasing excess nutrients through their waste right next to plant roots. So there's this cycle, this biological cycle going on. By the way, a cycle that we completely disrupt when we use a lot of modern agricultural practices. Protozoa are one of the first things to disappear with a lot of fertilization, the kind of fertilization that we use commercially in a lot of agricultural practices. And so then the existence of these protozoa and nematodes, their presence in the soil, since we can see them on a microscope, they're key indicators to us. Do we have a problem? Do we have a toxic residue from where someone used to spray herbicides on this field? Are the protozoa and nematodes back? Okay, we're going in the right direction. We've got the things that tell us that the ecosystem is recovering. This is a beautiful picture from, I believe, one of our former students, fungal hyphae. And this would be a basidiomycete, right, Elaine? Because that clamp connection, we know the kind of fungi that it is.
Probably
So,
one of the best fungi that you can have for most crops.
Right. This is just a beautiful clarity, and it shows you that if there are millions of these filaments in your soil, and they're connected to plants, and they're being fed, and they're helping to give the plants nutrients, your soil is being entangled by all those filaments. It's hard for us to have, I mean, it reduces the capacity for erosion, because the particles would actually have to get ripped apart, right, by something before they would become unstable. So,
And those aggregates are going to hold water, so that you don't have to irrigate in the dry part of the year just before you're going to harvest. So there's the structure that is built by these organisms is just amazing, and how much more you retain in the soil. Leaching is not going to happen. The nutrients in that leachate are going to be held in the soil instead of being lost to the soil. And so why do you need inorganic fertilizers? If you're keeping all the nutrients in the soil, none of them escape, none of them leave, because you've got well-structured soil built, thank you, by bacteria, fungi, protozoa, nematodes, microarthropods, earthworms, incatriads, all those good things.
Wow, that you're getting good at saying all those organisms at once. And you know what? There's only about 1,000 more to learn. So, okay, video's worth 1,000 words. This is something we love to see in soil, because we know that it's consuming those bacteria, and it's releasing a lot of nutrients right near the roots of our plants. So this is a beautiful little testate amoeba. It's going to come crawling across the surface using its pseudopodia, multiple. And notice it's got like a little shell. Reminds me of a hermit crab, kind of. To help protect it, it can go back inside there. Okay, we gotta see if we can get the slide to go forward. This is a fungal feeding nematode. Elaine, tell us why we know it's a fungal feeding nematode.
Well, we want to look at what's up in its mouth. So you can see the spear. It's kind of hard to point to, and the organism is wiggling. But you see that strand moving, yeah, and it'll be easier to point these things out. At the bottom of that spear, there are a pair of there could be a pair of knobs. And if you see a pair of knobs at the end of that spear, then it's a root feeder. But there are no knobs at the end of that spear. And so it's a fungal feeder. And you need these guys because as those fungi grow and hold on to all those nutrients, sequester and keep all those nutrients within its body, you need to have a predator come along and at least eat some of that fungal biomass, releasing those nutrients right in the root system of the plant. So the plant doesn't have to work that hard in order to get the nutrients it needs to grow. So here you can see where it stopped. You can see the spear right up there. There are no knobs in here. If there were some knobs, there would be knobs sitting here on the edges of the spear, but they're not there. And therefore, this is a highly beneficial nutrient cycler that we want to have in our soils. And especially as you go from bacterial dominated soil into fungal dominated soil, you want the predators that are going to be able to release the sets of nutrients that are being held inside the bodies of the bacteria and the fungi and the protozoa and the nematodes.
Perfect. Beautiful. Okay. This one is just fun, because they're so cute. So everybody likes a water bear, which is this little guy, a tardigrade.
This guy. You can see its little feet hanging. It looks like a roly-poly of a kind.
And then this other organism crawling around on its surface that almost looks like it's cleaning it is a rotifer, which is another multicellular organism that we see in soil. Oh, go ahead.
You can see the little rotors. The cilia are on the end of the rotifer, and that's the brush that it uses to scrape the bacteria off the body of the tardigrade. And you can see how much the tardigrade's just kind of leaning back, going, "Oh yeah, getting all spruced up for my Saturday night association that I've got to go to." So yeah, it's fun to make up kind of stories about what's going on with these guys.
Well, the other thing that we know is similar with rotifers, ciliates, which if we see a few, not to worry. Soil is and compost, it's not homogeneous. There's a place for there to be some little pockets of lower oxygen and all, and they help to feed on the things that we wouldn't want to grow out of control. They're they don't scare me. But if you see an explosion of ciliates, like in this video, that may start to tell you something's going a little haywire here, right? If we saw this in teas or something, we would say, why is there no competition with flagellates and amoebae in here? Why is, why are these things drawn? They seem to be drawn to pockets of low oxygen, especially when we're brewing teas. And so they're indicators again. They themselves, we're not going to judge them as good guys, bad guys, all of that, but we're going to say, what are they telling us, right?
What are they telling us about the habitat, about the environment? And we've got to have it promoting the beneficial organisms instead of promoting the anaerobic requiring organisms that are present in your soil.
Exactly. So these relationships, the picture of the soil food web is really the cornerstone, the keystone, again, of what we teach here at the school, and the approach that Dr. Elaine has developed over time, that we would understand who's missing, how do we get them back, how do we produce these composts that have diverse and abundant individuals, populations of all these different key beneficial groups. How do we apply that effectively in our farm system? The right timing, we can put certain amendments on leaves to help protect them from being chewed on by insects. I just saw a presentation this morning from one of our students that with a couple of applications of extract per year, she boosted her protozoa population substantially and really got a much better nutrient cycling outcome for her crop. And then we're going back to the monitoring piece. You know, this is where we start to know what the health, not just of the physical components of soil or the chemical components, which has been focused on for years, but to know the biological health, that holistic soil food web and what that means. And so you can look on our YouTube channel and see where we've gone through these components in more detail. You know, today is much more of that broad overview. But I just want to emphasize again, and I'm about done with my slides, and you'll hear more from Dr. Elaine about those higher level functions that happen when you have healthy soil. But what we have here is this beautiful loop, or this connection between plants and microorganisms. And I studied this with mycorrhizal fungi and plants in grad school. Some people are calling this the holobiont framework, right? That the plant is making sugars, releasing them into its root system, feeding microorganisms in various ways, helping to select which ones that it really needs or wants there. They're returning more nutrition, stress tolerance, water, all of these other things through their activities that help the plant achieve really optimized levels of photosynthesis so that it has more sugars to share. And so it grows more microorganisms, and then it has more sugars to share. It's a cycle. And unfortunately, in most industrial agricultural settings, we've broken these relationships, right? The practices, the tillage, the fertilization has actually disrupted this beautiful systematic regeneration and function that can happen. And so, Elaine, tell the people what happens when you break the soil food web.
If you destroy those organisms in your soil that have been working with your plants for the last billion years to cycle nutrients, to prevent diseases and pests from being able to attack that plant, building soil structure so root systems can go down as deep as they're supposed to go down, so that you have all the nutrients being taken up. When you destroy those interactions, the plants that you want are not going to be able to protect themselves. They're not going to get the nutrients they need. You eat that plant material, but it doesn't taste good at all, because flavor is what determines what is determined by the proper balance of all of the nutrients inside that plant, not just nitrogen, not just phosphorus. It's got to be properly balanced through the whole nutrient network. So there are some 40, perhaps a few more very important nutrients. You don't need lots of them, but you need some. So can your plant live without arsenic? Actually, no, it can't. There's a certain amount of arsenic that's required, not very much. Well, can you go over the edge? Sure. Too much arsenic is going to kill you. So how do you know where that balance is? And most of the plants that we grow, we don't know what that balance is. And yet you've got to have those nutrients. You need all of them. So we want to make certain that we're not killing those organisms that perform these functions for your plant. We need to make certain that we have the mineral nutrients inside the plant so it can be full production for us to be eating it. It'll have all the nutrients that we need in order to be healthy. We don't want to be using inorganic fertilizers, because every inorganic fertilizer is a salt. And so any inorganic fertilizer applied is going to be taking some water away from your microorganisms, and of course too, from your plants. Pesticides are toxic chemicals. Some of them are so toxic that you have to wear a complete moon suit in order to go out and apply that pesticide. That's how toxic it is. And yet we think that that's okay, that we're not going to be consuming some of that toxic chemical that's a residue left over on the plant. What decomposes those pesticides? What gets rid of the excess pesticide that's applied? It's biology, except you just killed them. And you killed more of them with the inorganic fertilizers. And now you've got diseases that run rampant. Erosion, where those nutrients moved into the soil, applied to the soil surface in many instances. There's no way for dirt to hold on to those nutrients. And so erosion occurs. Your soil is not held in place. It washes down the hill, and then it causes problems for every ecosystem that it moves through. We need to realize this, and we need to stop doing this, because Mother Nature already has a really great system. You don't have to do much work. You need only to be checking and making certain that the biology in your soil is what your plant requires. So think about, if you didn't have to put on inorganic fertilizers, how much money would you save? So stick that in the plus column. You're going to save that much. And typically on 1 acre of land, on general, for most of the crops we grow, you will save $600 per acre by not applying those inorganic fertilizers. You do have to make sure you get the biology into the soil, so that normal natural nitrogen cycling, nutrient cycling happens for your plant. So we've got...
Elaine, I'm sorry to interrupt. We've lost your camera. Would you mind trying to switch it back on if you can, please?
Okay, where did it go?
While you're looking for that, Elaine, I've also got the next slide is a video that I think would be a great place to show it here with what you're talking about.
Okay.
You're all good, Elaine. We can see you now.
Okay, great.
How does nutrient cycling work? During photosynthesis, plants combine carbon dioxide from the atmosphere and sunlight to produce simple sugars and carbohydrates. But just like humans, plants can't live on sugars and carbs alone. Fortunately for plants, there's a rich source of nutrients right under their feet in the soil parent material and in organic matter. When dead plant material, or organic matter, falls to the surface of the soil, it's gradually decomposed by microorganisms. Nutrients are then released into the soil in plant-available form. The next generation of plants then absorbs these nutrients, and so nutrient cycling begins again. We humans remove nutrients from this cycle when we take food from our fields, but these are easily replenished when microorganisms harvest nutrients from the parent material, the rocks and pebbles, and the much smaller particles such as sand, silts, and clays. At the molecular level, the parent material comprises large lattice structures which hold nitrogen, phosphorus, potassium, boron, calcium, iron, and all the other nutrients that a plant needs. With a balanced soil food web in place, plants can control the nutrient cycling that's happening in the root zone. They invest some of the sugars and carbs they produce during photosynthesis into the soil in order to feed bacteria and fungi. This causes bacterial and fungal populations to increase dramatically. The bacteria and fungi then get busy harvesting nutrients from the organic matter and parent material, absorbing these nutrients into their bodies. Predatory microorganisms are attracted, and they begin consuming the bacteria and fungi. The wastes left behind by these predators contains an abundance of nutrients in plant-available form. These are easily absorbed into the plant roots. So the plant gets a good return on its investment, exchanging sugars and carbs in return for all the other nutrients it requires. This results in well-nourished, resilient plants and in nutrient-dense food for us humans. With a balanced soil food web in place, plants are able to access all the nutrients they need, on demand, simply by varying the compounds they release into the soil. This means that farmers don't need to waste money buying and applying fertilizers. The soil food web costs very little to maintain by comparison. Having nutrient-rich plants results in greater yields and improved quality of the produce. Reduced costs and greater yields result in dramatically increasing profits for many farmers who have used the soil food web approach. The nitrogen-based fertilizers that farmers use are made using huge amounts of fossil fuels. Only around 40% of these inorganic fertilizers get absorbed by crops. The rest is either volatilized, polluting the atmosphere, or it runs off polluting our rivers, lakes, and oceans, causing algal blooms and dead zones that are a major environmental problem. So restoring the soil food web leads to nutrient-rich food for the consumer, increased profits for the farmer, and also benefits the environment. For more information about the numerous benefits of the soil food web and how you can get involved, please visit soilfoodweb.com.
Okay, and we have a few more slides here, Elaine, but we probably have to kick it into high gear because I'm sure that the people want to hear Ronald's presentation as well.
Well, so we have this list of regenerated soil functions where we're going to protect so much. We can't go through everything on every organism. Yeah, true. And I was getting a little deep, I guess. But yeah, so go to the cartoons that we have, like the one we just watched, that explain about making nutrients available, about building soil structure. Next slide. Doing the job of getting rid of this. This is what happens when you don't have the organisms in the soil performing all of the functions. We'll have a fire or something, or a flood, and all of the plant material is dead, it's gone, nothing to prevent severe drying problems. Highly compacted soil here, no water, no ability for roots to get to the water it needs. It doesn't retain nutrients. So you have runoff, you have leaching, and how do you reverse this? You've got to get the biology back into the soil. We want to make certain that the plant has the balance of all the nutrients it needs, because when that plant is then attacked by diseases or pests, that triggers the immune system of the plant. If you don't have enough nutrients, there's no immune system for your plant to turn on. And so you get high levels of diseases and pest problems. Keep going. So, here and dig and join in, Adam, if you want. But the claims are that growing sustainable means lower yields, more weeds, poor nutrition. Only if you have no conception of what soil is supposed to be. Soil has to have the organic matter. It's got to have the organisms, because the organisms need something to eat. That you need that organic matter. And you've got to have these organisms, predator-prey relations going on to release the nutrients that are not plant available in a mineral form in your soil. So if you don't resuscitate your organisms, then this claim that going sustainable means lower yields, more weeds, poor nutrition, yes, this will happen. But you got to build soil, not dirt. You've got to get the organisms back into that soil to do the jobs.
That was probably one of my biggest mindset shifts when I came to work for the school. As I said, oh my goodness, like 90% of what's out there in the research, the land that's being managed, they're teaching people how to be quote unquote successful dirt farmers, right? By putting all these inputs in year after year, not thinking about the soil as alive, but thinking about, well, we just want to extract and exploit and pull food out and sell it and make a profit. And that's what it's all focused on. Meanwhile, we're destroying the planet.
Yep. So when somebody says, oh, Dr. Ingham, she doesn't know what she's doing because I tried that compost stuff. Well, okay, where are your numbers for the biology? And they look at you like, there's something wrong with your brain. We put compost out that I wouldn't call it compost. They put putrefied organic matter out. And of course, that's not going to promote the necessary organisms. So we need to know about the entire food web. And that's where you need to learn how to use a microscope, or develop a relationship with somebody who is doing the assessments of the microorganisms. If you've got somebody close by, It's you drive your samples to the lab and they start working on them, and they show you exactly what's been improved, what's been lost since the last time you looked at the biology. Does your plant have the nutrients? Is determined by whether you've got the organisms that do the nutrient cycling. So agriculture really has to focus on the soil biology. That's what's going to make things successful.
Absolutely. And that's why we love that this series has so many examples of people who are, ignoring the advice about sustainable or biological farming that is out there, and they're actually saying, wait, we made it happen, we made it work. Right? Where's all this advice from people saying, well, you just have to keep doing agriculture this way with chemicals, when in fact soil biology, it's proving, people like Ronald are proving that soil biology can feed the world, can keep people employed in agriculture. What do you think about Earth Day, Elaine?
Yeah, I think we need to have not just one day a year for Earth, for having the soil. It's got to be every day, because you eat every day. You better be making certain that the cycling that nature is so good at performing and has been managing to do for the last billion years with plants, with roots. But before that, of course, everything started with bacteria 4.5 billion years ago. Fungi, 3 to 3.5 billion years ago. And what was the interaction then with the bacteria and the fungi? Well, and then we start getting the predators into the system, and then you start getting predators of the predators. This whole system works together so amazingly well. And yet it's what we're destroying faster than anything else on this planet.
Mm-hmm.
Why does this not compute?
We're getting there. I have to remind myself we're making progress, because I see it with our students. And well, I've stopped sharing so that Ronald can take over and he can show us some of the progress that's being made in the world.
Thank you, Adam. Thank you, Elaine, for great information. And Ronald, take it away. And Ronald, I think you're on mute. Oh, still on mute, Ronald?
Yeah.
There
No,
you go.
probably.
Great.
Just like being a little stressed out. Oh, this is my time. I have to be ready. Oh
The
my
spotlight's
God.
on you.
Right. Thank you, Brian. Right. So let's go. So I already introduced myself. Thank you, Adam. Thank you, Elaine. Most specifically Elaine, because you're the reason why I'm doing this today, and I'm really grateful for that. And hopefully this talk will also help a lot of people around the audience to feel inspired, because this work, this stuff, this methodology, this science works. And I'm going to try to share what I do and how we do it. Indeed, our mission statement at Fluorescence is the following. Beyond my previous background in finance, I had made my choice a couple of years ago, and this is about being on the forefront, making the thing happen. All the concept that has been mentioned just previously is about how this actually function for real. So this is what we do. We enable farmers to make transition. Usually when I start the conversation with a farmer, there's always this touchy moment about, oh yeah, how much will that cost? So, well, you guys may be expensive. So it's about valuing soil health. And I asked them, how many years have you been putting NPK in your soil, right? So just basically we could take the 60 last years. And I make some research, and the FAO, as Adam was mentioning, the UN group for agriculture, has the number up there, and this is the number, right? It's in trillions, millions, tons, right? So this is not even computable with money. So if anyone has data and and numbers about how much did cost the NPK during all those years. Well, give it to me. We'll have fun making numbers, but it's not even computable. So why don't you just invest 2 years in a regenerative program, right? Maybe the soil deserves it. Maybe you deserve it because of the amount of money you wasted to actually degenerate the soil. So continuing on, how do we do that? This is a little bit of photos about what we do. From compost tea, from those compost teas in the field, working cover crops, no tilling, organizing the transition, teaching the farmers the microbiology, and most recently now we're starting on fungal farming. We've just teamed up with a company in Peru that is providing some setups for large-scale fungal farming. Continuing on here, this is the fundamental of what we do because those two things are correlated. Soil microbiology implies nutrient cycling, implies plant health, productivity, and carbon more stable in your soil. You probably have heard that it's all about, the Paris Agreement and how many carbon in the air and how can we sequester that? Well, guess what? There is a huge amount of lands where humans are doing agriculture, and this is how we can sequester more carbon. So regenerating using microbiology is a way to have carbon back in the soil on a stable form. Sorry, I'm going fast. So we have been working recently on Havana tobacco. We're proud to say this is the first regenerative tobacco worldwide. Again, well, in the definition that we have when we say regenerative. 2 parts for that presentation. We're going to focus on the first one because I have little time. Sorry, I'm rushing because there is a lot of info. We're going to cover all those fundamental points that you need to address when you actually regenerate. Fertilizers, microbiology, no-till, pest, and yields. And the second part will correlate the same findings with nutrient cycling, nutrients, because this is all the farmer thinks about, nutrients, how much do I have to put N, P, and K, the pH, of course, and then a beautiful story of phytotoxicity remediation, a story that Paul Stamets will absolutely enjoy. And we are proud to say that microbiology helped that. And we're going to see that in the second part at later in time for with other webinar. And then a bit of ecosystem, which means how can we create farms our ecosystem. So here are the 7 principles of soil health that is pretty much widely shared among the regenerative community. It's pretty much standard. We talk about microbiology, cover plants all year, enjoying the whole thing, and if you can have livestock, good. And the rest, the point 1 and 7, it's about humans, the hardest path actually to deal with. So a little bit of background: tobacco smoking, well, comes back from South America, Central America, North America. It's a sacred plant. It has been used for curing people forever, and it's kind of fun that today it turned out in that it's a huge market. It's a huge market with a lot of a limited amount of players with a lot of money involved, and most of the producing countries are this one. And this experience that I'm sharing is based in Central America. So pick your country. You may find where I worked that out. Anyway, continuing on here, we are dealing with an industry which is really highly workforce intense, very low mechanization, and we indeed enjoyed the fact that because you have so many hands working that out, well, you could enjoy to be really more precise in terms of what you want to do for caring those plants. Because when you do agriculture, you don't grow a field, you grow plants, right? And so if you can care plant by plant, well, just, let's do it. And we enjoyed it because you don't have 2 chances to do a first impression. And so we just went full ballistic and used the workforce as it was offered. So here the context was the following. We've been transplanting the seedlings before the normal period. These people had an organic regime. We're going to see a little bit what is that about. We had double the amount of rainfall. So in Central America, you can imagine what can that look like. It was kind of a real challenge. but we made it. And finally, we had to compare to conventional because too much rain, they decided to come back to conventional. And so that was even better for us because we were full regenerative and we could compare to the actual conventional regime. And this whole trial is a 60 days harvest from transplant to harvest. So here are the main part in details. Sorry, again, I'm going fast. We're going to deepen a little bit more for each of these points. Basically, talking about fertilizer, microbiology, compaction, pests, and yields, all what we've just talked with Adam and Elaine in practice here with tobacco for cigar making. So the first work you want to look at when you go regenerative, it's fertilizer, right? Even though they said they were going regenerative, they had their program, which was using some of these, organic products that are fertilizers, supposedly, but they are just like greenwashed, right? Here is the kind of product that they use, and if you pay a close look, if the definition is enough, you can see that, well, it's kind of pretty conventional way of, advertising the product, okay? So then you have NPK in the central on the orange box, and then you have another NPK in the black bottle, and then you have magnesium and potassium on the right, and then on the left you have U-mix coming from Leonardite, which is a product that is extracted with a massive caustic thing, which leaves nothing living in there. So when they said, okay, this is our organic regime, and I said, are you seriously telling me that this is organic? Well, so of course they didn't answer that. This is broadly what they were applying in their organic product. And on our side, we have been having our tools, our beautiful compost. And here, the luck that we have, these people were already applying the Soil Food Web standards in terms of compost and vermicompost. So we used all that we had on the top of some bokashi. Some of you know a little bit of bokashi, and biol, which is a biofermented liquid that pretty widespread in South America, and some bioagents. So yes, we didn't put any of their commercial inputs there because for us, these type of organic products are just a joke in terms of organic agriculture, as they call it. So we've used these types of compost, and his has been the sequence roughly that we have applied to make this magic happen. Because when you think about it, well, are you seriously telling me that these microbes makes the nutrient available for these plants? Are you serious? Well, duh. Yes, absolutely. So solid compost extracts with bioagents, meaning some organic nitrogen in there, which means like oils, hydrolysates, flour of peanuts and stuff like that, algae and stuff, and also a foliar work. Continuing here are the numbers. A conventional regime costs you that much. An organic regime with this product that I just showed costs that much. This is how much we cost: 0. So the most people in the audience that are a little bit, let's say, money-oriented or cost-oriented should tell me, yes, but you didn't input the cost of the compost itself, compost making. It's true. However, this is an absolutely fantastic achievement to say we have used 0. I can remember the owner when I brought him to the field, he looked at those leaves and he said in Spanish, salvaje, which means wild. And we're going to see that the result in the end. Talking about microbiology, what we've just covered, who does what and why. We have some ratios, we have some benchmarks, and from those benchmarks, we've been increasing these in less than 60 days. It's about taking this theoretical concept where we have a succession going on with a relationship in biomass between fungi and bacteria, giving us this F2B ratio. And here, okay, from this context, we've decided where we placed the tobacco. And so that was our target. And also replicating a less fancy graph than Adam showed, and I actually would have to have your slide, Adam, if you want to share it with me, about the trophic level, which means who's eating who. It's the poop loop story, as someone I know is saying. It's like all the waste that one just leaves going to be plant available. How does that translate? It translates in this kind of somehow undigestible numbers, but I'm going to explain that very fast because my time is counted here. So all the stars are the numbers which are representing our targets at the different trophic levels, which means all the functional groups that are interacting, eating each other. And here we're comparing these two, which is the control plot. It's a conventional plot in orange, 52 days after transplant, and ours, the regenerative field here, which has the data that we can compare. If we look at the ratio of the fungal to bacterial ratio, all the percentages are on the right. We are 50% up in less than 50 days after transplant. Then bacterial number, roughly same. We won't enter detail here, but fungal level already 70% up. On the pathogenic side of things, when we talk about omycetes, for example, that we know most omycetes are pathogenic for plants when you have no soil structure, when you have no diversity of roots, when you have all those type of things, when you have no water infiltration correctly made by the rest that I just mentioned, well, then you have a lot more of omycetes because they are kind of the water mold things here. So less of these as well, minus 38%. And then the protozoa that Adam was mentioning, well, we are skyrocketing above them. And here, at the last part, the orange block here, in terms of nematodes, we have presence of the bacterial feeding nematode, which is a sign that our nutrient cycling is started, which means the plant can actually be fed by the microbiology itself, right? Because if you don't have all that, well, of course, you're going to have poor health and poor yields in your plant, right? Because plants need microbiology. They co-evolve together. That's the truth. Now, talking about rhizosphere depth, or in other words, compaction. We've been deepening the compaction layer of more than 178%, is what Adam was mentioning about his client in South Dakota, if I got it right. Well, how did we do it? Well, we have been first asked if we wanted to plow. Why? No way. What did we do? We used the local tools. So yes, horses, we just worked the beds. The client has already put a cover plant type of strategy here. And so we decided not to destroy it because the plant was already doing the work for it. And you can see next to the arrow where the horse is, well, we still see the grasses. Anyway, here, we measured the methodology here. We know that plants can't push their roots beyond 150 psi. So we measured at which depth our plot was going in comparison to their plots, right? And the 300 one is like just for fun. So here are the two cases. On the left, this is our plot. On the right, this is theirs, which means the control, which means the conventional, which means plowed, and then herbicides and all the whole 9 yards, right? So we are reaching 42 centimeters depths where the plant is, well, where we reaching 150 psi, they reach, on their side, it's only 15, right? So the numbers are just simple. We have been nearly multiplying by 200 in less than 60 days. Okay, the second part of this presentation will explain how. Now, talking about pests, we just talked about it. Incidence of total number of pests, which means how many different types of pests have been appearing during the whole 60 days. Not the importance of it, just the numbers of those pests. Well, this is our monitoring board, and you can count the green boxes. For our side, 1, 2, 3, 4, 5, 7, and this is the number of pests that we have been seeing coming up to our field. A little detail here is these two numbers is like when the farmer decided to chop the rest of the surrounding field that was having cover crop, suddenly the aphids exploded. Anyway, coming back to ours, 7 total. Now this is theirs. You can count, it's 11. Okay, 11 to 7, it's a good decrease. It's a 37% decrease. And here too, the two red blocks show the same relationship, the same dates. The farmers has killed all the cover crops around, and they also had a massive bounce in the aphid incidence on their field. Anyway, it's an observation. Now, in terms of costs, minus 20% cost, because we have been working on a sort of, how can I say, it's kind of an opportunistic way. You have a list of product and that, target crops, and some product target some special pests, then some cost more than others. So you just switch and adjust the whole thing. It's a cost efficiency thing. We had to play it because we wanted to make an impact, and it worked. And also because we had, as you've seen, the whole this is the whole sequence. When the pest was not reaching the orange spot, well, we wouldn't apply. That's just this. You have the mindset of preventive. Farmer always goes preventive. Sometimes you have to be bold enough and say, well, I have to take the risk to show if it works or not. And that time we did it, and that's what makes this story a success. So that's how much a phytosanitary regime cost, and that's how much we did have to spend if we had to spend the money. I mean, they spent it. Talking about yields, before the actual finance harvest, we felt it was interesting to share and to tell them what was the achievement for our trial, because you can imagine when you do a regeneration program, it's kind of, you are a show in the farm, and even more when it's a big farm. So we measured height and harvestable leaf number. For their standards, it was 15 each long, right? So that's us at field at that very moment. Then the results are the following. Our average is on the left, 74 centimeters height with an average harvestable leaves of 6.4. On their side, a higher plant, but 6.6, 0.2 difference of harvest. Are you serious? Is that really meaningful? Yes, it's meaningful. This is a regenerative tobacco, right? Same yield. And so then we went just up until harvest, and now it's just the agronomist that has been looking at it and evaluating what we had. And this is what it looks like in the background, right? This is the regenerative tobacco here. It's 1 ton per unit of land, 0.7 here, 65 days after transplant. And this is what theirs look like at 55 days. So it's not like at pure maturity, but you can see a difference already. And they yield 1 ton, 1.1, like 50 kg more, right? Are you serious? Right. It's meaningful. Yes. So this is the result of all this trial. We can call that a success. It's a success, no doubt. It's doable. We still have some work to do in terms of the management and all that. And it's kind of our mission to just tell you that, yes, this stuff functions, right? And I'm done.
Got to put myself off mute. Thank you, Ronald. Awesome to see all the different data points, and really, I mean, your farmer's got to have a big smile on their face. Yeah, very, very happy with those results. It's always great. I feel that kind of symbiosis there when the farmers start to see, like, oh my gosh, there's some huge changes. Like my farmer Jen is going to be presenting in webinar 3. She was blown away about the trussing that her tomatoes did. Never seen anything like that before. She's like,
yeah,
and so
it's
vigorous.
fun because tomato is the same family as tobacco, by the way.
Exactly. Yep. Okay, let's go ahead and we're going to talk about the promotion. So give me 1 second while I share up my slide. And okay, we're going to talk about our January promotion, and we have a small video that we're going to share here in just a second. We do have a promotion going on our BioComplete Compost Bundle, which is really going to be geared towards farmers, ranchers, and growers. And then we have the Consultants Kickstarter Bundle, which is going to be for people that want to do work like Ronald and myself do, consultants out in the field. This is going to be the final time that we're going to have such a significant discount at 45% off. So I just want to make sure that everybody's aware that the January promotion is going to be, I think, the last of our really big discounts. And let's go ahead and talk or see a video about the bundles.
As far as I know, there's not a lot of people out there on this planet that has collectively looked at all these different sciences and said, this is basically how it works. that what we call the soil food web, this predator-prey interaction. It's so complex, yet really so basic. It just makes a lot of sense. I wanted to know what the problem was out in the field. A lot of times plants were sick. Either it was a nutritional problem, or it was an insect or a disease problem. And instead of just treating it with a 911 chemical pesticide or fertilizer, I wanted to know what the underlying cause was. And the underlying cause was something to do with the soil. And it just made so much sense when I learned from Dr. Elaine Ingham what the missing link, the catalyst that made organic fertilizers and amendments work. It really wasn't the product as much as it was the biology, if it was there in the soil.
There's organic and then there's biological. And you really cannot have a successful organic or conventional growing system without the active biology. And soil degradation over the last years is one of the top environmental issues. So having as many people as we can bringing attention to this issue, I think is really important.
Where they say, without GMO and conventional farming, we would not be able to feed the world, that's totally untrue. Because the data is there on what we do when it comes to growing plants regeneratively, because of the savings, they're enormous, and the increase in yields are huge. You can actually back that up with the science behind soil food web and quantifying the microbes. It's really more than just the course. It's actually a organization of like-minded people that are making a major change to the environment, to our food supply, and you can continue to learn, take more courses, and advance into more of a role of an advisor over time. But that is a process, it really is. We're always learning as a group, as a science, and it's a great collective group of people around the country and overseas that we all kind of communicate with each other.
It's like you're a soil explorer that gets to look at a whole world that a lot of people just walk over every day and don't think much about.
One of my clients out in the Midwest does soy and corn. In 3 years, they completely eliminated Roundup. They have switched over to non-GMO, and just that alone has saved them over $1 million. Most conventional farmers in soy and corn, for example, they're going to spend between $800 and $1,000 an acre, their cost. Where this particular client is probably spending $150 to $200 an acre now. And now we're sourcing out local resources that at one time would have gone to the probably local dump. Now it's going to their farm, and we're recycling it and making high-end compost out of it, and then also making liquid extracts out of it on a much larger scale. And now they're increasing their yields. Our objective for this particular client, he's looking to get over 300 to 350 bushels an acre versus the conventional 170 to 200. They see a lot more life. They're seeing butterflies and frogs and salamanders and snakes, and all these things have disappeared from their environment. Now they're coming back. And I think that's really satisfying. What business can you go into, make money, but yet you're giving back to society, you're giving back to the environment, and you're giving back to your children?
If you're interested in joining the soil revolution, this is a great time. We're making the BioComplete Compost Bundle available for the final time at the price of $38.70. With the BioComplete Compost Bundle, you can register for the Soil Food Web Foundation courses with Dr. Elaine Ingham for just $38.70, saving over $1,100 through January 26th. You'll also get Stage 1 of the Grower Training Program totally free, saving a further $15.40. That's 26 hours of mentor time dedicated to helping you make your own biological compost and develop your microscopy skills to qualify as a certified Soil Food Web lab technician. You'll also get 2 free bonuses with this offer: the Introduction to Permaculture course by Graham Bell and the Soil Sponge Regeneration Workshop with Dee Dee Purshouse, saving a further $500. The total value of this bundle is over $7,000, for which you'll pay only $38.70, saving over $3,100. That's 45% off. Remember, this is the final time that the BioComplete Compost Bundle will be made available at this price, and there are limited places available, so please don't delay.
Okay, let's go ahead and jump into our Q&A. So panelists, if you can, go ahead and take yourself off of mute. That'd be fantastic. And we're going to spend the rest of the webinar actually answering questions that you guys had submitted. So, and thank you. I've been watching the Q&A board. There's a lot of fantastic questions in there. We're only going to get to a fraction of those, but it's great to see all the interest in the unique questions that everybody's generating. And this question comes from Cindy. And Cindy asks, when cover cropping, can a seed mixture be inoculated before seeding with compost tea or worm tea, thus only using machinery on a field once versus twice and still achieving the same effect? What say you?
Absolutely. We can do
Absolutely.
this. So we want to, usually when you're dealing with your grower, they don't want the wetness of the seeds in the container, because the seeds then stick together and you're putting 2 or 3 seeds down at a single time. So you want to either put such a small amount of the compost extract or tea, spritz it really lightly where the seeds have the time to dry out before they go into the seed bin, or you're going to want to soak for a minute or two. And a lot of the things that we're seeing is if you've got a good way to maintain movement of the water around the seeds, that you don't have to hold your seeds in that liquid for very long, so 30 seconds instead of 15 minutes. So there's no fear of the seeds germinating because they got totally involved, total amount of water that they needed in order to germinate. So get the seeds into the liquid for a short period of time, pull it out, dry the seed down, and then that can be stored for quite some time as well. So it's always, go ahead in January, February, when you have nothing else to do, right? It's go ahead and do the seed coating, and it works really well, putting it out in the field.
Yeah, I'm a huge fan of, at the time of planting, anytime you can introduce biology during planting. And when those seeds do imbibe, there's microbiology right there ready to start that interaction. And you can really see the response in the plants. And I also think the germination rates go up as well, that they have much more success and vigor from the get-go. And I would say, on a large scale, there's some of the challenges. You have to use a seed drill. And again, Millie mentioned wet seeds don't go through seed drills very well. There are seed drills that have sprays at the bottom, so when the seed drops, it gets coated when it's going through. But I would have to say, for your home gardeners, for the people that are going to be just planting for themselves, I like to actually soak my seeds in compost extract or tea for 24 hours and let them imbibe, really get ready to germinate and plant them right away. And you're hand planting, so you don't need to worry about a seed drill. And it works fantastic that way too. So
when we work in some agricultural fields, people will put in a furrow, and then they will drizzle the compost extract in the furrow right on the surface of the soil. And then the next thing that drops in to that furrow is your seed, right on top of a really good mixture of those microorganisms. You close up the furrow, and germination is reduced by maybe a week, 10 days. And you don't have to be going back in with a second application of something, or worrying about getting your seeds to imbibe, whatever. Couple ways to approach this particular question.
For sure.
I can add up here beyond what you guys just said. I also on the dryness topic, when you coat your seeds, you can also get the very fine dust of your compost that it's like right there on your soil or whatever, and you can just drizzle that over your seeds and just mix it up again, and then if you want to really go push it forward, you can also take some of the bioagents, so like powdered fish hydrolyzate or algae, and then you can just drizzle like this all over the seeds, and then your seeds are coated with microbiology, with the food for the microbiology and the nutrients for the microbiology, and then you drill it. Would that be large scale or small scale? And most farmers have the tools actually.
Yeah.
To actually
Already.
coat the seeds, right?
Mhm.
So it's
We aren't going to make you buy more and new equipment. We'll make
No,
do
it's
with
it.
what you already have.
Yeah.
Exactly. Adam, Carla, anything you guys want to add?
Oh, yes, pretty much that. Thank you.
Okay, great.
Yeah.
All right, let's go ahead and jump to the next question. Let me share up my screen again. Okay, question number 2. This one's from Bonnie, and the question is, can soil pH be altered by increasing the soil microbiome, or are soil mineral additives needed? What say you, panelists?
Elaine taught me this one.
Ooh,
Yes. I came in thinking, because I went to a land grant university and all, I was like, well, you've got to actually get the chemistry altered with lime or something, right? And then she helped me to understand that, like, the things that the microbes are producing, the glues and all, can alter that soil pH, as well as, sometimes there can be a real benefit to bringing in the right kinds of organic amendments, right? But we should think of these things much more like, can we do it once and then let the microbes do it the rest of the time, right? Like Dee Dee has said many times on previous webinars, we should think of the microorganisms as the essential workforce of the biome of this planet. And humans, we have a role. We are part of nature, but we don't have to micromanage everything in there and constantly be thinking about adjusting pH. Plants can also somewhat adjust their habitat and pH with the kinds of acids that they produce. So trust the soil food web. This is what Dr. Elaine has slowly gotten into my skull.
Just to complement, remember that we need to work with local soil biology, right? Don't bring compost from other country or from another biome, because first, the biology might not survive, it's not adapted to your conditions. Second, it takes much longer for you to see any benefit. You have to apply much more amendments. So always work with local. Did I learn right, Dr. Elaine?
You get a star. Ding, ding, ding,
Yeah, I mean, I can add up on these questions, on the 2 parts of the question, actually, from that trial. I didn't show the data because it's for part 2, but for the same time frame, we have the data. We have moved the pH in 60 days just with the microbiology. It's just incredible. I mean, I had a hard time when I first heard from Elaine, oh yeah, let the plant do her work alone. You have a multiplicity, diversity of pH around the rhizosphere. And I was like, are you serious? And yes, well, I just proved it to myself. And about nutrients, adding nutrients. Okay, that's the big topic. What I can say about that, it's also what type of availability of nutrients you bring back to the soil. You don't have the microbiology, well, you don't make anything available to the plant, right? And what type of compost are you putting in? A very young compost or very mature, a vermicompost, and all those data we could double check and say, yes, that's true. Plants need nutrients, microbiology, and organic matter. So then the whole thing functions on its own. Guess what? This is Mother Nature design. It was all sorted out to start with.
Yeah, there's the graphic that Adam presented with the keystone. And
Absolutely.
it always really, it hits back to me that in a lot of traditional agronomy, I look at there's 3 principles of soil. There's the structure, the physical, the chemistry, and the biology. And so much of today's agrochemical farming is based upon the structure and the chemistry. And they're going to use a lot of chemical means or mechanical means to influence those 2 properties of soil. When in reality, biology is going to influence those 2 areas very significantly. So if your focus is on biology, you're going to impact both the physicality and the chemistry of the soil. And that just hits home for me. Sorry. Go ahead, Carla.
Surprisingly, this same logic applies in the majority of ecological restoration projects so far. They're also starting to realize that it's not just the biology we can see. All this biology that's in soil are vital to have any successional restoration project going on. So the word is coming out and the system is changing, so which is good.
It's great.
There's
Elaine,
a metaphor.
oh, go ahead, Adam.
Oh,
Sorry.
I'm sorry. I just wanted to say there's a metaphor that I like here because I love metaphors. If you're trying to aim at a target, do you want a small target or a big target to hit, right? The bullseye can be bigger if you have sufficient organic matter and all these microorganisms in your soil. It just makes it easier to hit everything, every target, every optimal thing in soil when you focus on the biology.
Yep.
Costs less.
Yeah, for sure. All those things. Okay, anything the panelists want to add to this question before we move on to the next one? All right, let's move to the next question. Okay, so this one's from Peggy, and Peggy asks, how much will nematodes and microarthropods retard or prevent development of mycorrhizal networks by consuming fungi? Well, I think one of us can answer that question very effectively.
I mean, I think a lot of times we think of competition as a negative, but competition is part of how nature shapes these systems. And there are mycorrhizal fungi that I've studied, the arbuscular mycorrhizal fungi, that actually don't help plants that much, right? And so I want there to be competition in there. I want there to be like a little bit of stress for everything. I want everything, because we tend to see that when we make it easy for something, we artificially make it easy, that it can actually become kind of like a cheater in these systems. So like, also with all fungi, the thing that we would emphasize is like, we don't want to see an overgrowth and dominance of one species in there. We want diversity. So I think of these little fungal feeding nematodes and microarthropods as pruners of those fungi. They're pruning it. They're helping, maybe even stimulating more growth in the long run, right? There's so much that we don't understand, but it's not like mycorrhizae good, therefore what hurts mycorrhizae is bad. It's everything needs to be in these relationships. And I mean, it's hard. It's a different way of thinking about the world. I wish the answers were easier, but nature does not owe it to us to give us easy answers.
It's complex. Well, it reminds me of the predator-prey relationships. That any successful ecosystem needs to have things in balance. You need to have predators to be able to keep the prey populations in check, and vice versa. If you have too much predators, then you're going to reduce the ability of the prey species, and so you have to have balance, and that's what we're trying to strive for, is kind of the ecological balance.
Yeah.
Yeah.
And
To
I
get
want to add,
things
yeah,
to
Elaine,
get
go ahead.
to get things going and get up to speed and let them do all of the different jobs that they do for the plant. So how often do we go through and say, well, this fungus does process number 1, 2, 3, 4, 5, and 6. Well, this guy only does 1 and 2. This one does 3 and 5. We don't know that number 1 is the one that we want to promote and maintain. We haven't even begun to consider that part of the interaction that goes on in soil. So we have only just begun to understand this, and there's so much more that we can do to improve plant production, reduce costs, make it better food for us and our animals. We've got, come on, guys, get to work.
You got it.
Yeah, and I wanted to just share my own experience here, because when I first started and looked at the benchmark and how much of this, how much of that, and okay, you need to go and get there, and then suddenly you wake up and you go the epiphany moment, like, but it's all dynamic. And then it's like, this group eats maybe that group, and that group will regrow, and that group will be eaten by this other group, and this other group, and this other group. It's like it's dynamic. which is why the whole framework that we have is so important, because it shows soil health measure as presence of things that contributes to plant health or yield or productivity or whatever you want to name it. So it's dynamic, all that, which means, okay, we can measure into the nitty-gritty of, how much of this and that, but at the end of the day, we are limited, right? We know that the presents are good, so we want to have the whole soil food web and mycorrhizal. Yeah, it's another of those that we need in the system.
Life is a constant revolution, right?
Revolution, evolution. Yep. Okay. Anything else, panelists, to add before we move on to the next question? Okay. Next question it is. This question's from Anonymous. The question is, where do you get the proper soil biology to add to degraded soil? What say you, panelists?
Well, you go out into whatever ecosystem you have in your biome that hasn't been disturbed much, that hasn't gotten pesticides or other toxic chemicals, inorganic fertilizers applied to it, and you collect some of that to use as an inoculum. You don't need even a pound if you're making a single bed of compost when you're first getting started, because you always want to do practice first, because if you don't put things in the proper balance, you're going to have extreme temperatures happen, or nothing ever happens, or it dries out too quick. It doesn't. So you're going to be taking test piles, and that's a real good place to be applying the inoculum of, just what fits in your hands, and mix that into the compost, so that you get started on this process of maximizing the diversity. Well, if you go out once a year and grab a handful, you're not getting the full gamut of all of the different species. So you want to go out at every change of the season. Every time this plant comes into the system, you should get a little bit of that inoculum of the soil and inoculate it into your compost piles. Anything that you can think of that there's something different about this soil or this plant, this community, you want to be putting in those organisms that changed the environment. So this other species of plant or other set of organisms could now be leading the charge. And you just want to maximize diversity, because like Ronald was saying here, we don't know exactly, we can't isolate them, we can't grow them on a petri dish. You've got to go to the soil. Don't buy materials. I think Carla said, don't buy stuff from a biome that's from far, far away. It's there are quite a few examples of people who isolated out really good sets of bacteria or fungi and put them all into a container, and in the greenhouse, whoa, the plants just grew like crazy. But you take that material out into the real world and it doesn't do a thing, because limited species are not capable of tolerating the changes in temperature, moisture, the mixture of all of that. They can't stand it when they get peed on by a cow or something else. So maximize diversity. You want to get out there and do that, and remember to go out and get inoculums from different parts of your biome and on different dates within your biome.
And once you have all this good local biology, keep that pile as a mother pile, so you can build more piles and use inoculum from that original one, so you can multiply the biology. And also we have to, like Dr. Lane said brilliantly, we have to take care to not get on the loop of the biological egg, but just changing from chemical to the biological, just using a few commercial inoculum. We're not going to do anything in the long run. So use your critical thinking and really question every single step along the way.
Yeah. In compost, and we've mentioned this a few times around compost, there is, I think, people take compost in a very generic sense and say all compost is equal, and it's really, really not the case. There are composts that are really full of bad organisms that aren't really well suited to growing the cash crops we're trying to grow, all the way to the spectrum that it's extraordinarily biologically rich. And so really understanding the compost that you're using, does it have the necessary biology, is a key thing, because really compost is the basic or the base for a lot of the work that we do. That's how we inoculate the soils with the good biology, is through the use of something like compost or vermicompost. And so if you're going to be using compost or vermicompost, just make sure it has biology. That's such a critical thing. All right, panelists, anything else you want to add to this?
I'll just say again a metaphor. Sometimes those commercial products make me think someone's like, I'm really interested in birds and I want to have a lot of diverse birds, so I'm releasing chickens into my environment.
It's a bird. Exactly. That's too funny. I'll have to use that one next time, Adam. Thanks. Okay, let's jump to the next question then. So this question is from Jay. And Jay says, how does worm castings compare to regular compost? Does it have the same / similar microorganisms? Would it have any fungal presence? What say you, panelists?
Oh yeah, worm castings are really good if you do the work necessary to get the diversity of microorganisms into that composting process. Worms will chow down by taking a bite out of whatever is in front of it, basically, and then works on it all the way through its digestive system. The rear end of the worm is a place where the worm itself got the mix of bacteria, fungi, protozoa, nematodes, growing in its own digestive system, got it from mom. So mom transfers to all the babes the inoculum of organisms that are really good. Well, if she was bringing along bad guy, excuse me for my simplicity in expressing that, bad guys, disease causers, causing problems with the growth of the plant, if the mama earthworm was carrying that, she'd die, and of course there would be no more offspring. So though in general, very good inoculum occurs in the worms. And so even if you're going to do a thermal pile, maybe you'd want to go out to really healthy places in your biome and grab an earthworm or two and stick that into your pile, because you're adding that inoculum of really good things. Like Carla said, have a pile that you are putting all these interesting things into the system, all beneficial. Well, and that means you've got to keep that pile aerobic. You have to make certain that you are not putting in anything that selects for bad guy organisms versus the good guys. So you can generalize and think along those lines. Now, how do you know that the organisms that are in your pile are any good? How do you know if they're going to be good guys or they're going to be bad guys? And that's where you have to have a microscope, because you can't see it in any other way, that you've got all of the different bacterial species that's possible under your conditions. The fungi that you see are a good mix of all the beneficials. You want to know that the protozoa that you've got in there are the flagellates and the amoebae. You really don't want to be seeing a boatload of ciliates. One or two, as Adam said at the beginning, one or two, not a problem. Don't worry about it. It's when you've got the mass conglomeration and it's just, oh goodness, something very unhappy is happening in that pile, and the ciliates are trying to tell you. Pay attention to messages from Mother Nature. So same thing with nematodes. If all you've got is a herd of root-feeding nematodes, you're not going to have plants in your field pretty quickly. So take a take a look at what's present. You want bacterial feeding nematodes, you want fungal feeding nematodes, and you want nematodes that attack and consume the root feeding nematodes. Their favorite food most of the time are root feeding nematodes. So let's have a boatload of those in your piles as well. So select for the beneficials and maintain that, and then allow things like, some of the other critters, microarthropods, macroarthropods, birds, people always get all concerned about birds landing on the surface of the pile and pooping. Oh my gosh, all the bad guys are in that bird poop. No, don't think so. Only under strange conditions would you have to really get serious about that. And because we are managing the sets of organisms who are present in our compost or in our soil, we're managing the habitat so that we're selecting for the beneficials and against the detrimentals, against the diseases and pests.
Thank you. I want to add a little thing here. Again, coming from this recent trial that I showed up, the second part is dealing with that. The main source of our extract was from vermicompost, just that, right? So then we're not going into detail of how much of the nutrient are made available, what are the extra compounds that are in the worm cast, which is the vermicompost that we use, that are beneficial and protecting the rhizosphere. Well, think about it, how nature does it, right? When you go to the forest, what if you just dig a little bit, you'll see some worms. What are they doing? Okay, they're just taking up and down things, like bringing some of the nutrients back and up, and then make it available for your plants. Yes, there is a difference in quality when you use these type of young compost or one that has been through the worm intestines. For me, this is the Rolls Royce. If you can use vermicompost to your regeneration, you win it. That's simple as that. And I have the data that can show that.
Yeah, I love worms. They are just our allies. I mean, they're really in the trenches with us. And it's funny, almost every single compost pile I've ever built, like a hand turn pile, has always turned into a vermicompost pile. I can't keep the worms out. They just show up and they start doing what they're doing. And there's a lot of people too that will make a thermophilic compost, which is really we're making compost using heat, moisture, and time, and kind of turning on a regular schedule. And then they'll take the finished thermophilic compost and add it to vermicompost bins to do additional processing, to break down that material into finer and finer particles. Worms are essential allies to us. Gotta love the worms.
Yes, someone asked in the chat, how can we attract worms if we can only find the commercial ones that are not local? I mean, you can try, it's not a promise, but have a pile of dry leaves or wood sticks and get that in a cage. They will show up in the bottom of this material. This is how I started my worm bin. So worth the try.
One thing that attracts the earthworms is the moisture. You really want the moisture to be somewhere around 60%, maybe 70%. So, it doesn't do much good to go out to somebody's lawn and try to dig up and find earthworms in there, because it's just way too dry. It's too compacted. So think about what habitat is right to promote those wanted organisms, and then go and put the earthworms in there. Then when you go to utilize that material, you might want to sieve out the earthworms, so that you're not sending the earthworms off to someplace. You want to keep them on in your own piles and use your worms to enhance your compost. Maybe you'd sell a box of earthworms to every person you sold a ton or more of compost, because then they could maintain these really good organisms. It takes so long to make a worm compost pile, to get it finished, that it takes somewhere on the 6 months to a year to start from scratch and build a worm bin. Once you're there, it only requires, you're still at 3 months to 3 to 6 months to make a really, and see, and I'm not a patient person. So, where I'm concerned, I will throw a few earthworms in and they'll go with the pile. But I like thermal compost because it only takes 21 to 28 days.
That's funny, Elaine. I'm not patient.
I'm not
I want
patient.
results now.
Instantaneously.
She's pretty patient with me.
Yeah. You're not a worm, I guess.
I want to say one more thing about worms, which is there are parts of the world where worms are not, like, in the recent time frame, part of the ecosystems, and we can see invasive worms being a problem in some forests and other things. So I want people to be very cautious about what they might be releasing into their environment, and to do some research on, similar to what Carla said about just bringing a bug in a jug product from somewhere else. If you live in northern Mexico and you get worms from the Pacific Northwest, I'm not even sure they're going to do very well.
Right.
But there's also the fear that they could become like Wormzilla out there and cause some kind of problem. So just let's be cautious about that aspect of moving biology around, because I think by some accounts there are like 50,000 non-native species in North America, and some of them are fine, but some of them are devastating to biodiversity.
You might want to take a look at what certain earthworms will do. So, for example, the work that was done up in Saskatchewan, it's at the University of Saskatoon. They noticed that the conifer forests were starting to die off, have a lot of problems. And what was the problem? All kind of research was going on. And what they discovered was that the lumbricus, night crawlers that came in to the lakes, with fishermen. And at the end of the day, when the fishermen were done fishing, they would take their jug of earthworms and throw it out into the soil in that pine forest area, thinking that they were doing good things for the soil. This just made it better. Well, no, because lumbricus comes up to the surface of the soil and grabs the organic matter off the surface of the soil and brings it down into their burrow and uses it to make a fungal garden that then the earthworm eats that wonderful fungus. But up above, it's changing the way that fungal to bacterial biomass ratio when you have a lot of that particular kind of earthworm, because they select for bacteria and against fungi. And so the forests were being wiped out by something that everybody thought as being a really good earthworm. So what do you now have to add into that forest system? To get rid of the lumbricus. Well, there you've got to go searching for the appropriate mycorrhizal fungus to get growing on those root systems again and inoculate something that is going to be competitive with lumbricus. So there is a solution, but sometimes it takes years to get some of these things figured out. So what's the safer way to approach all of this? Make certain that you've gotten at least a handful or two of the ecosystem or that part in your biome that you're going to be selling to your growers. Won't that be fun? We got to know everything.
For sure.
Life is too short.
It is.
But we've improved so much in the last, 40 years.
Beauty of teamwork as well, right? Don't work alone. Find your peers and develop together.
Yep. And keep your eyes and ears open for when somebody says, oh, we've got a problem starting to develop. Well, then maybe we can be the people who really help fix that problem right from the beginning The answer will include the biology.
Okay, I think we have time for one more question. So let's take a question from Dylan. And Dylan, is no-till farming still one of the best practices, or have new methods been developed?
Absolutely. No-till farming, actual true no-till farming is the best practice, but it may take you a little bit of time. You know, if you go look in your soil and use your microscope to figure out what biology is in there, and you have no bacteria, you have no fungi, you have no protozoa, you have no nematodes, there are no earthworms, that is not soil, that is dead dirt. And you can't call it anything else. So now, how are you going to prepare your dirt so that you can get the best effect of the additions of the compost and the extracts and whatever however you're going to apply that biology? You are going to till one last time to break up the compaction that is absolutely 100% going to be the problem. So get in there, get into the soil, put maybe a quarter, half an inch of your compost on the surface of the soil, and then till that in. Yes, you're going to be killing organisms in that inoculum that you're adding in, but it's better than nothing. So 50% of the organisms are going to be killed by the tillage that you do to mix those organisms into the soil. But then you'll come back a week later and find and do another microscope test. Who survived, who didn't? So you have to put another application in. You might, are you going to add in a solid, or you're going to add in a liquid? And this is where you have a consultant, that you want to have a consultant helping you interpret what those soil biology tests are telling you, so that you can, as rapidly as possible, get your soil back into a condition that's going to promote the growth of the plant that you want.
Yeah, I think no-till is essential, and really, it's kind of funny. It's not that the practices are changing. There's just a lot of different techniques. I mean, one of the main reasons why people till is for weed suppression, and that's the thing we have to really, help those farmers with, is how do we suppress those weeds as we're transforming that property. I mean, for me, nirvana is if I can get a perennial cover plants growing in those systems that are already highly competing against those weeds. The soil biome has changed so that it supports more mid-successional types of plants versus those early successional weedy species that tend to be a very big problem in a lot of agriculture. And that's just the techniques that we use. How do we do that weed suppression until we can kind of get that system into balance? And there's a lot of different techniques. There are a whole bunch of different ways upon how you can do that weed suppression. Panelists, anything else you guys want to add?
This is not my bit of poetry, but I'm going to steal it or remind us of it, that when sunlight is transformed by plants and turned into sugars, we can think of the exudates of the plants as liquid sunlight, right?
What?
Liquid sunlight. And when that liquid sunlight goes into the soil, it will do the most to bring that soil alive if we don't let it get out too quickly, which is one of the things that tillage does, right? You chop and you mix all that oxygen in there, and those bacteria start going bananas and having a feeding frenzy, and then they breathe a lot, and then a lot of that CO2 is lost. Oh, you also kill your fungi a lot. You chop them up when you till. And so that liquid sunlight, or those connections to plant roots that the fungi have, that would have put that carbon, that liquid sunlight, into a more stable form. It would have slowed its rate of decomposition because of the way that fungal cell walls are. Instead, when you till, you're putting it into the biological pathway through the bacteria, and it's very inefficient. It goes back into the atmosphere. And so, sometimes people charge agriculture with something between 15 and 25% of global greenhouse gas emissions, and people fight. It depends on the country, it depends on the system, all of that. People fight about what exactly that is. Some people claim as high as 50%. But I say none of that matters, because agriculture can be the solution to so much carbon in our atmosphere, right? And so many of these other greenhouse gases. If we can take that and, using the magic of plants, get that liquid sunlight to stay in the soil longer, which is why we wouldn't till, which is why we would bring back these microorganisms that can store longer, like 5%, 6%, 7% organic matter. We're seeing this on farms that are transitioning to soil biology instead of a measly.
And on large scale farms. So
Right.
it's possible, doesn't matter your scale.
Yeah.
Yeah. Historically, the soils held 5 times the carbon of the atmosphere. And a lot of the greenhouse gases we have up in the atmosphere right now are because of historic use of tillage.
Yeah.
Even back in ancient Rome.
Yep.
So let's hold the liquid sunlight in the soil.
Agreed. Okay, panelists, anything else you want to add before we close? Okay.
Do we have an extra hour?
Yeah, exactly. It feels like it's always just too short. All of us are talkers. We can definitely talk this through. Okay, well, I'd like to be able to go ahead and close our webinar for today. And before we close, I just want to give a reminder to the 3 other webinars that we have coming up in our Soil Food Web Movement webinar series. So again, webinar 2 is Wednesday, January 18th, and this is going to be talking to the Soil Food Web community. And it's really going to be talking to the consultants, the people out there doing this kind of work to help make that transformation. And then webinar 3 is going to be a success story, and I'm going to be having one of my clients, Jen Serp from Sonoterra Farms, talking about her successes that she has with her farm. And then and that's Thursday, January 26th. And then webinar 4 is going to be on Tuesday, January 31st. And that's going to be Grass Valley Farms in Montana. And Casey Ernst is going to be talking with Corey Miller. So look forward to seeing all you there for our next 3 webinars. And with that, I would like to go ahead and just thank all of the people that work behind the scenes to help these webinars go. There's a team of folks that do a fantastic job of getting all the promotion material out there, get the webinars up and running. Without them, we would not be able to do these webinars. So a big round of applause out to our support team that really makes this happen. And I'd like to be able to thank our panelists today for being able to share their information. It's fantastic to share our knowledge and to answer questions from the community. And I'd like to end with thanking Dr. Elaine Ingham for all the work that she's done. She's been an inspiration to us all, and it really, really has helped, I think, make this work a possibility. So thank you all, and we'll see you on the next webinar, everybody.
Thank you for hosting,
Don't
Brian.
forget to click that like button, subscribe to our channel, and ring the notification bell to stay updated with all our new videos.
I'm a talker, so.
You always make so much sense.